Okay, so what we have here is a T cell, and there are CD8 T cells called cytotoxic killer cells, and there are CD4 T cells, which are helper T cells. The CD8 cytotoxic killer cells present CD8 cluster differentiation 8, which binds to an MHC1. MHC1 stands for Major Histocompatibility Complex, and MHC1 is the type of MHC that is presented on any and all nucleated cells in the body. So basically anything but a red blood cell, probably. And MHC2 is only presented by antigen presenting cells and B cells, B lymphocytes. The MHC1 is composed of an alpha chain, and it can fit a amino acid chain in here, protein on it, that is 8 to 11 amino acids long, which is shorter than MHC2, which can fit one that's 10 to 30 amino acids long. The MHC1 is associated, the alpha chain is associated with a beta chain, but they're not covalently bound. This is beta 2 microglobulin, and this fits in there. Whereas the MHC class 2 is composed of an alpha and a beta chain that are both bound to the membrane. This one's not, over here it's not bound to the membrane. These are both bound to the membrane, and they are non-covalently associated with each other. There are the A, B, and C genes for MHC1, and there is the DQ, DR, and DP genes that code for MHC2. The DQ gene has a certain manifestation, DQ 3.2, that is seen in most everybody who has diabetes. It doesn't mean that if you have it that you'll get diabetes, but of people that do have diabetes, usually they all have this gene, which results in them essentially phagocytosing their own insulin or something like that. There are typically 6 to 8 genes that encode for MHC2, and that results in 11 to 12 different kinds of MHC2 that can be displayed in a single person. MHC1 and MHC2 are codominantly expressed from the parents, when an MHC binds to a CD4, or I'll put it this way, basically the only way that this MHC can present a antigen, this is MHC2 in this case, I guess, an antigen to this guy and have it receive it, is if they're bound together. Like this won't, if this MHC wasn't there, this would not bind right there. At the same time as this MHC or CD4, CD8 presentation, there also has to be the CD28 and the B7, CD28 being on the T cell and B7 being on the antigen presenting cell. That's a co-stimulator, and if that is not present, then this won't happen. When T cells get activated through this process, they typically release IFN gamma, which causes differentiation of macrophages. The way in which the MHC ends up being presented in the case of a MHC1 is typically there will be these cytosolic proteins that will be inside the cell, and at one point they'll become broken up and ubiquinated, so they'll have this little ubiquinone attached to them. So this little piece of the protein and the ubiquinone will go in through this TAP tap, which allows it into the ER. And at
at some point that tap will close. I believe it's through Tapicin. I think that's what Tapicin does, is closes the TAP. And there is this ERAP that knocks off this little thing off of the, knocks off the UB, I think, off of the something, off of the cytosolic protein that's been broken up. At which point the cytosolic protein can bind to the MHC1 within the ER, and then it can kind of pinch off and then go to the outer membrane, plasma membrane, where it can present with its little piece of cytosolic protein, at which point would be when like a T cell would come along and bind and recognize it. A CD8 cell would do that with MHC1. If you're looking at an antigen-presenting cell or B cell, what happens is they're, rather than getting their inner cytosolic proteins or whatever, they are eating up external proteins. So this would be some kind of protein antigen floating around in the fluid of the body. And they're going to eat this, and they're going to break it up into little pieces. And at the same time, you have in the ER, the cell is producing MHC2, which has this little clip thing in it, and an invariant chain. The HLA-DM, I believe, breaks apart this invariant chain and removes the clip out of there, and then the MHC2 binds to the protein antigen, and then it is then manifested on the outside of the cell, MHC2 pigmentation. And that is the majority of the lecture.